Our last speaker is Hongyi from Stockholm University. Sorry if I misspelled your name.
That's very good, thank you. First of all, I would like to thank the organizers for giving us the opportunity to share our latest development in MicroED, which is using electron diffraction to study macromolecular crystals. And I think it's a very good organization that we're starting the day with MicroED, now we're ending the day with MicroED. And having listened to the previous talk and MicroED, hopefully by the end I can convince you it's also a very powerful home source that you can use to study your protein crystals. Right, so I'd like to start with the acknowledgement. So this is the group I work in. So Xiaodong is the large group leader, and people with the name highlighted, they work with me on the macromolecular crystallography, and they are all in the audience. So if you have any questions later, you can also talk with them. And most of the work I'm going to present here is done actually by them. And also we have our key collaborators from the biochemistry, biophysics department at Stockholm University in Martin Högblom's group. And also we work very closely with the cryo-EM facility in Sweden. So that's the SciLife lab at Stockholm. Okay, so a quick introduction of our group. So we've been working on electron crystallography for the past 30 years. So we started with imaging, combining images of very thin crystals with diffraction. And then we moved on to 3D electron diffraction or MicroED data collection about 12 years ago. And we also developed a lot of softwares trying to help electron microscopists and crystallographers to collect MicroED data on their microscopes. Right, and for the past 12 years, we mainly focused on material science. So we work with a lot of small molecules like porous materials, zeolite, metal-organic frameworks, and other kind of crystalline materials that we can put into the microscope and collect diffraction data for. And since 2015, after the first paper of the MicroED came out, we started to work with protein crystals as well as the small organic molecules. So there really has been quite a lot of development in this area internationally and also in our lab. So hopefully I can show you why it's a quite nice method. So the first thing that I would like to say is that MicroED or 3D ED is complementary to all the other techniques. So some people may tell you that it's going to solve water problems, which is not true. Which method to use really depends on your problem. Right, and also because of the development of X-ray crystallography and also single particle cryo-EM, we're actually learning a lot from these fields, and that's why MicroED has been developed really quickly in recent years. And one of the major reasons we want to use electron as the diffraction source is that we can work with much smaller crystals because of the strong interaction between electron and your crystal. So couple of talks ago, you heard that the speaker has been struggling with these needle crystals, right, in this droplet. So we actually love this kind of crystals. So all these large single crystals you can use for X-ray is way too large for us. And we actually work with these very thin needle crystals. And I can show you that how easily we can collect data from them. Right, and another reason we want to use electron is that electron interacts with electron cloud as well as the nuclei, so it's in between X-ray and also neutron diffraction. So in principle, we can study the charge of ions in the structure directly. But this is in principle, and there has been a proof of principle paper, which I'll come back to a little bit later to say why I'm emphasizing the in principle at the moment. And also we have dynamical effect, and you heard there are a lot of errors that in X-ray diffraction. I think in electron diffraction we have even more errors, and one of those was dynamical effect, which is due to the strong interaction between electrons and the crystal. So electrons can get scattered multiple times going through a crystal. So this causes the intensity we measure become really inaccurate compared to X-ray. Right, but now there are a lot of efforts being put into this to model this dynamical effect, and by doing this, we can actually get more information from the crystal. And last but not least, electrons can be focused very easily, so we can form very nice images of the crystal, or we can even take high resolution images, atomic resolution images of the crystal. In this case, the material science, but we can also take images of protein crystals as well. Right, so I just want to give you one example of why MicroED really makes when MicroED really makes the difference. So this is a protein that is responsible for the immune signaling in the body. So the crystal actually forms by just mixing two proteins together, which is the MAO-TA domain with the MyD88 TA domain. So our collaborators actually been working on this with XFEL for some time before they came to us. And the problem they had is that when they have collected the diffraction data from these crystals, it was very difficult to index them. And until we put in these crystals into the microscope, we realized that these thin crystals actually bundle together. They form like a stack of thin needle crystals. So the diffraction pattern you get from these crystals will be very difficult to index because they came from multiple crystals. But with the capability of doing imaging in a microscope, we can look for single crystals and then collect the rotation 3D data from these single crystals. And then those crystals actually diffracted beautifully. And by doing this, we can solve this very interesting structure, which is one of the only few new structures of MicroED. And there was actually an X-ray structure of this same protein, but this was in monomer form, which is crystallized using X-ray crystallography methods. And with the MicroED structures, it's really what's happened, how the protein works in the body, that it dimerizes, and it really gives a lot of biological insights to our collaborators. Right, and these are all the crystals we've worked with. It's a gallery of things. So you see that we love needles, especially the sea urchin needles, because we can get lots of crystals on the grid, and we can, in principle, collect the 3D data from each one of them. Right. We also have crystal fragments and also stacked plates, and we can break them into like individual plates and then collect very nice data from this. And actually, MicroED now is a standard service at a Swedish cryo-EM facility at SciLife Lab, and I believe it's also service at EBIC at the Diamond Light Source. So please give it a try if you have seen these kind of crystals in the crystallization plates. And because we're collecting data in a TEM, in order to have a electron beam, we have to have relatively high vacuum environment. So this means we have to prepare the sample in a similar way as cryo-EM people do, right? To vitrify the sample so that the crystals is not destroyed by the vacuum. And there are several ways to do it. And actually, it's not so difficult anymore that you can do pretty much each one of them and then just vitrify the protein crystal and put it into TM. And then if the crystal size is suitable, we can put it into a microscope directly to measure. And if the crystals are slightly too large, there has been development of FIB milling, which is putting this into a SEM and using gallium iron or other irons to mill a thin lamella from the crystal. And this has very particular use in some cases. And from Tamir Gonen's lab in the US, they've shown that it's very useful to study membrane proteins growing LCP, for example. These are very viscous mother liquor. And then this lipids, and then they could mill a crystal lamella from these membrane proteins and collect very nice data from them. And also recently, they've shown that by doing this, they could collect very high resolution data as well. So this is really optimization of sample preparation, right? But the problem is that this, what they collected was 0.9 angstrom. They could do direct method, direct facing with this data, but this is triclinical lysozyme. For X-ray, the resolution is 0.5. So the take-home message is if you can do X-ray, if you have large enough crystals, still collect them at a synchrotron or home source, you're still going to get better data by having X-ray. And we are taking a slightly different approach, and hopefully it become obvious a little bit later why we do this. And because it's a crystallography method, so we thought to optimize the crystallization condition is another approach, and it's actually easier than doing a cryo-FIB, because for cryo-FIB you spend maybe half an hour to mill just one crystal and it needs a lot of expertise. for de-optimization, going backwards from what you usually do for X-ray crystallography is that it doesn't take very long to actually produce these kind of small crystals in a suspension. And that's how we do it. It's a brilliant idea came up by Gary, and the idea is very simple, very similar to the seeding procedures you do for X-ray crystallography, right? You put in some seeds and then use the vortex and some beads to mill it into very small seeds. And then we add back some protein into the drop and then hopefully grow some protein crystals at the same time as the next round of milling. So you're milling into large crystals into small seeds at the same time, growing the crystals and recovering the packing in a way. And then we ended up with the very, very small crystals. And that's what we see on the grid. So we've tried this, Gary has tried this with multiple examples. Most of them non-standard, non-model protein. And usually what we get is just like a milky looking suspension. And if we can see crystals under the microscope, but they're already too large under the optical microscope. So we really want to see this kind of consistency at the end of this process. And what we can then have is on a typical TEM grid, which has about 200 to 300 mesh. And from each mesh, we have tens of crystals in principle that can be used for single crystal data collection using the rotation method. Right, so this is one of the examples he did recently. It's a non-model protein, and the resolution, you see, it can be very high because we optimized the sizes of the crystal and the packing as well. And this is the map that came with this kind of data. So this was very nice. So comparing to X-ray, I think for the same structure, X-ray had a resolution of around 1 angstrom. So we are still a little bit behind, but I think we are getting quite close in this way. And now I'm going to switch gear a little bit. So why do we bother to develop this method, or we want to use MicroED? So the first reason is that, as I said, we can work with much smaller crystals. So you've seen one example of the MIDI88. And recently, I think by the EBIC group, they've shown a very nice work that they could study those crystals actually grown inside the cell. So these are microcrystals grown in the cell. And they could collect 3D electron diffraction data or MicroED data directly from these crystals, which is very nice. And another reason is that, as I said, we could probe the charge state of ions, hopefully in the near future, in your macromolecular structures. And thirdly, we can do hopefully high throughput ligand screening, right? Because we have so many crystals now on the grid, we're thinking about ways to how to improve the threshold to do ligand screening or fragment screening. And last but not least, there are also possibilities to do room temperature measurements. So I'm going to touch upon this a little bit later. So this is one of the projects Laura has been working on, which is refine the charge directly. So in principle, as I said, the scattering factors of ions, electron scattering factors, actually show quite large differences at low resolution range. So in principle, when she did the simulation, which is calculating the structure factors and then the electrostatic potential map from models with different charged ions, in principle, if we do simulated data, we could see a huge difference actually between these charged ions. Right, and then the problem is that if we can collect data from them, right? So these are actually the crystals our collaborator used for XFEL experiment, and we could collect the electron diffraction data up to 5 angstrom from this crystal, and they are slightly too large for us. So using Gary's method, we can produce this very nice thin plate or needle-like, ribbon-like crystals, and this is what resolution we can get. But after playing with the data, I think we see more problems than getting answers. So there are 3 major problems. First, it's very fundamental. So how much electron dose we can give to the crystal before the charge gets reduced, which is, I think, very important question. And I think the physics behind electron diffraction and X-ray diffraction is slightly different, and all these secondary electrons generated by electrons, because the crystal is so small, so maybe they escape much easily, cause less reduction of the ions. So that's something we are hoping, and we're working on electron energy loss spectroscopy, trying to figure out what is the critical dose that is going to cause the reduction of the charge. And secondly, the isolated spherical model of the electron scattering factor is not really accurate for doing this kind of experiments, right? So we really have to consider the scattering factor of the ions in a chemical environment. So that's something we're also trying to figure out and working with some of the experts in the field. And thirdly, as you can see here, we have severe beam damage. So if we just take one diffraction shot from the crystal, we can get 1.5 angstrom resolution data. But if we take 3D data collection, we cannot get this resolution simply because of beam damage. So we are now trying to design new ways, maybe doing serial electron diffraction, or doing a small wedge data collection, or doing helical scan on these crystals. Right. Next, the protein ligand interaction. We had a preliminary study on this already a couple of years ago, and resolution was okay, and it's relatively quick, but this is a very simple system. We know everything is going to work if we've done things right. And currently what we are trying to achieve is higher resolution, better models, better crystals to do this, and also higher completeness, and also much higher throughput. And this is very good when we have lots of crystals on the grid. So now we are working with experts in nanofluidic handling. So we want to print different fragments on different areas of the grid. As you can see that then we can have different, hopefully binding interactions on different areas of the TM grid. And next is the nanofluidic channel. We are working with the DTU, a group in DTU, and these are actually TM holders, and you can put these nanofluidic chips into a vacuum environment. And the good thing about this is that we can pump in liquid while we are observing, we are collecting data. We can also put in gas when we are collecting data. And this is what some of the preliminary results look like. So this is not protein crystal yet, it's a small organic molecule. We actually were able to grow this crystal inside the channel. So this channel is very, very small, so 200 nanometers in width. And here is the crystal grown without any liquid. And then when we fill the liquid, you can see the contrast difference. And we were able to collect very nice MicroED data. And we got slightly higher background, but the data quality is quite nice, and we could solve the structure relatively easily. Okay, last but not least, I hope you are interested in the method now. And then the question is, can you do it at your at home? The answer is pretty much yes. Yeah. So we can do this on every microscope almost out there. So we've tried all these different microscopes. Those written in light blue are cryo-EMs. So you can talk with your cryo-EM facility and see if they're interested in collecting data for you. And we have also material science microscopes. Then we have to use a cryo-transfer holder. And for data collection, there are a lot of software out there. If you're into programming, you can use Instamatic, which is a Python open source package